So I will talk about the non-clinical development of advanced therapies and biologics. And what I meant to say is actually the last sentence on this slide, that especially with ATMPs and biologics, the regulatory framework, as you can imagine also from the talks in the first session, an ATMP and as well as a biologic is a very complex product, and each product is completely different. So it's quite challenging to cover all the requirements. Let's focus on safety requirements in this talk now. To cover that in a regulation or in a guideline or whatever is challenging. So what at least my experience in the last 12 years working as a consultant is that especially with this type of products, science is the basis for non-clinical development, and I will guide you through that. So usually the different types of products are covered in that scheme, and you might have seen similar pictures like this. Initially, I planned actually to compare or to present to you a non-clinical program that leads to a product in wound healing for each of the products, because my company was lucky enough was involved in a small molecule product, even in a herbal product, a peptide product, a cell-based product, and a non-standard biologic product, all in wound healing. And you can imagine that the programs look completely different. But I think that for this talk now, I focus on just comparing an ATMP and a biologic. And as you can imagine, and as we heard, I can skip that slide on the definition of a biologic product, as well as the slide on ATMPs. So there is a definition, and it's clear that it's quite detailed. But I promised to bring you back to science. I did not know that other people also had an elixir or a soup or whatever. I decided to present you that picture. But I think the statement on that graph is a little different. I think what is important also, if I have the chance to talk to scientists, I think it's very important that the step from pure scientific thinking into development is a very important step. And I work a lot with small biotech companies and academic groups, and even with new technologies like CRISPR-Cas, and you can imagine that a step you need in development, you need the scientists a lot, but you also have to adapt in which way you present science then later on to regulators. So there is a step, meaning that the people from development needs to go into science, but also scientists need to go out and think in the way of thinking of development. So, what is actually? Let's start at. I think there was a very nice interface at the end of the first session, when the non-clinical data, or actually, let's keep the right term, pharmacodynamic data of the ApoScience product was presented, or the secretome product was presented. This was actually how regulators would call the pharmacodynamic data set. That means that everything that has to do with the efficacy of the product, the mechanism of action, proof of concept in non-clinical animal models, things like that. So that's something which is important, and of course regulators and later on investigators need to make sure that any patient has the chance that the product really works in a specific dose. Let's skip the dose discussion for the meeting today. That's another very interesting topic for that type of products. That's one thing. And the other thing that each product needs to be defined in is safety. So you have for each product, independent on the nature of the product, be it a small molecule, a peptide, a secretome, a cell, you need to have, prior to initiating a clinical study, you need to have information on safety and potential efficacy. So there is sometimes a misunderstanding in terms of wording. So pharmacodynamics is everything that has to do with efficacy, and I think it's Dr. Osler just mentioned that it's that these gaps between having a scientific basis and initiating a study. You mentioned that in the context of product development. I'm mentioning this now in the context of pharmacodynamics, and I think we all should be very impressed how many data in terms of in vitro and in vivo data were presented on a specific product for a company that was mentioned before. But that is important, and it's also interesting to understand that for this part, the efficacy part in non-clinics, there is only very vague and not tight, but the opposite of tight guidelines, because even authorities cannot define what is needed for a specific product in a specific indication to allow for a clinical study. So that's always in the responsibility of the company. So, but if you allow for a gap, either if you want to save money, if you want to save time, or if there is no animal model, which can be, you might end up in speeding up a program, but you always end up in I usually call it the dark side of speeding up a program, because you go into a very expensive clinical study with much lower or with much less data that confirm that the chance of a successful development is there. So that's everything that has to do with efficacy is less regulated, but very important. So pharmacokinetics, just to mention this term, that everything, all data that has to do with examinations, what does the body with the product. So just for you to know, that's very easy to explain for a small molecule drug. So you take any type of product and you are interested what happens with the product in the body. Is it distributed to the liver? Is it excreted via urine? How long does it stay in the body? All those things. How does this translate into a secretome, for example? Is it to track the specific cytokines? Is it to what kind of constituent of that whole mixture of elixir constituents should be tracked, because we also learned that there are so many different mechanisms of action. So we have the vasodilation, we have the different mode of action, so which of the components is relevant for which. So it makes, of course, in that example, makes it very difficult to define what will be our pharmacokinetic data set that allows us to initiate a clinical study. So and then last but not least, the third component of a non-clinical testing program always has to do with safety, and you can imagine that that's the standard terms or the standard types of studies that are usually conducted with a product. But of course, if it doesn't make sense to do whatever a carcinogenicity study with a product that is only applied topically, for example, and never goes systemically, one would scientifically argue why it doesn't make sense to do one or the other study type. For example, genotoxicity. Is there any risk that the secretome has any genotoxic effect? Per se, I would say no, but you should not forget that it's a manufacturing process, so there might be an impurity from the manufacturing process. So that's always the question one asks. And so what I would like to do in the next 5 minutes is to just give you an example of two different products, a biologic and ATMP, similar to what Dr. Osler did for the CMC or for the product development, and to show you the differences. But it is very important to first understand that at least how we approach a product, a new product, is again via a risk-based approach, meaning that, so we learned in the first session, there is different mode of actions, including platelet aggregation and vasodilation. So that sounds positive in terms of efficacy, but platelet aggregation, couldn't it also be an adverse effect, which I don't like at a higher dose or in a different system? Same for vasodilation, there could always be a negative effect as well. So what we usually do when we start developing a product, or if we get involved in development of a product, we first define the risks. Then the second step is then to define which study could cover the risk. And then, of course, each study needs to end up in a whole program that at the end one can counter-check, can I address all risks in that program? Yeah. So, and that can of course, the risks depend on the indication, on the mode of action, on the type of product, and of course on the nature of the product. So what I will present here is an example of a biologic in the indication wound healing, but you can imagine there are so many different biologics, a monoclonal antibody, a peptide, and something so complex like a secretome, that all has different risks and therefore also might require a different approach. But now just coming back to my slides. So I've chosen the example of a biologic, which is a cell-based allergenic complex mix of components derived from human blood product formulated in hydrogel, and the ATMP, and these are just examples, so I'm not referring to a specific product. And another example would be an autologous stem cell product obtained from a skin biopsy. To make it a little bit more complex, the cells were selected for a specific marker with an antibody. And the different mechanism of action for the two products requires a single application for the ATMP, for the cell-based product, and requires repeated administration for the biologic. And first of all, one step back, you can imagine, I don't touch that now, but you can imagine that the first step is to define if it's a biologic or an ATMP, but we had this discussion before and I will skip that now. So, and as I said, please don't write it down as a standard program for an ATMP and for a biologic, but just as an example for a specific product in that product type. So, primary pharmacodynamic, I mentioned at the beginning that it's not covered in any guidance how you have to do an efficacy program. Every company needs to decide that by themselves. There is a little bit of guidance in some of the guidelines, but usually companies have to decide. Safety pharmacology means if one of the components or the component itself, if it's not the mixture, can induce any adverse effect on vital systems like the cardiovascular system, the central nervous system, or the gastrointestinal system, the renal system, just the physiological systems. And so it might be that there is a requirement for testing, since, don't forget, we have an example of a complex mixture. Could be that there are cytokines, whatever, in this mixture, so it might be of interest to at least exclude that some of the components have an adverse effect on central nervous system, for example. For a cell-based product, especially if it's applied on wounds, the probability that a cell or components of the cell from the surface wound goes into the system and induces some systemic adverse safety pharmacology effect is very low. So in a specific case, it can be that usually there is no safety pharmacology testing for a cell-based product. Then pharmacokinetics, mentioned before, it might well be that it makes sense to select specific components of a mixture as a lead parameter to see, do I have the same level of cytokines, for example, in the blood, or what is the level I'm ending up. That's related again with the CMC question. So how can we be sure that the batches that are used that the different batches are comparable? I did not say that there is a need in any case to do pharmacokinetic investigations of a complex mixture, but it might in some cases be useful just to have it as a monitoring, or just to also have some information on exposure. For a cell-based product, you would track cells usually, but with a completely different approach or question behind being, would a cell, especially if the cell were genetically modified, maybe distribute to other parts of the body, be it due to homing or whatever reasons, or genetically modification. And in the worst case, that's usually what authority want to exclude, that a cell or a modified cell go into the reproductive organs and have a reproductive risk for next generation. So that's the worst case. And so might well be that you need to select a scientific approach to track human cells or mouse cells in an animal model and to test if the cells persist. And then with toxicology, so depends of course on the intended mode of application in the clinics, and also on the limitations of an animal model. everyone can imagine that the application of a human cell into a mouse, assuming that there is a human cell, then into a mouse might lead this repeated application to adverse effects which are of no relevance at all for the clinical use. So even if it were of importance to have different routes of to have different applications, then the animal model might limit the application to a single dose. So in that case, one might think about repeated dosing in both cell products, but as I said, there are limitations. Genotoxicity, as I said before, if the manufacturing process doesn't bring any risk of other components into the product, a natural product usually is not considered of genotoxic risk. But of course, theoretically, there could be a tumorigenicity risk, meaning that a cell is either due to a donor issue, that the donor had a basic disease, or that the modification of a cell changes the cell in a way that there is a proliferation or tumorigenicity risk. Local tolerance is mostly related to vehicle, like for example a hydrogel which is already in use in other products, is of course of lower risk compared to if you develop a completely new vehicle. So that's the overview of, just to give you some insight how such a program could look like. And then I'm already actually at the take-home message. So what did I want to tell you with all that? So first of all, I think I hope that you get a little bit kind of misleading and misleaded in a way that you think, okay, now I don't know anything, because this I aim for, that you get the feeling there is not a standard program for an ATMP and not a standard program for a biologic. You always need to consider the nature of the product, meaning that you need to understand the science behind. And that's the need that in all those products, scientists need to be involved up to clinics and even later on. So that's my first take-home message. Second is safety testing of biologics is a little bit more regulated than safety testing of ATMPs. That has to do with the complexity of the program, but there are exemptions, as we heard. Then sometimes ATMP programs might appear smaller. That's because there are much more limitations in the availability of animal models. But first of all, there are exceptions, and second, might well be that the program is still much more expensive because you have the need for all that type of bioanalytical work. I mentioned that cells have to be tracked to exclude any persistence and migration of the cells. Then you need, of course, a number of analytical methods to be sure that you specifically track the cells that were applied, or to track the risk for tumorigenicity. All these artificial animal models, like immunocompromised models, at one point developed tumors by themselves, and of course you need to exclude, if you detect a tumor, is this tumor due to the product that I applied, or is it maybe a spontaneous tumor? So you also have to do a lot of analytical work to exclude that. And last but not least, I think we first of all have another session in the afternoon where we also have the really regulatory approach presented from regulators. And my recommendation and also my experience is that, especially with that type of product, one should approach regulatory authorities early on in development to discuss the deviation from any guideline, to discuss the scientific approach, to discuss if regulators and companies are of the same opinion in terms of interpreting risks. And so this, I would recommend everyone to take a chance of a scientific advice meeting or a pre-IND meeting in the US. And that's it.